Introduction to drug transport across membranes. Understanding how drugs cross biological membranes is crucial for determining their absorption, distribution, and effectiveness in the body. These membranes act as barriers, and drugs must navigate through them to reach their target sites. 1. Passive diffusion. Most drugs cross membranes via passive diffusion, moving from an area of high concentration to low concentration. This method doesn't require energy and is ideal for small, non-polar, lipid-soluble drugs. Example: diazepam, a lipid-soluble drug, easily passes through cell membranes via passive diffusion. 2. Facilitated diffusion. For drugs that aren't lipid-soluble, facilitated diffusion comes into play. Here, drugs move across membranes using carrier proteins or channels, but still follow the concentration gradient, so no energy is required. Example: glucose uses GLUT transporters to move into cells through facilitated diffusion. 3. Active transport. Active transport is necessary when drugs need to move against their concentration gradient from a low concentration to a high concentration. This process requires energy, typically in the form of ATP, and relies on specific carrier proteins. Example: levodopa is actively transported across the blood-brain barrier, allowing it to reach its site of action in the brain. 4. Endocytosis. In some cases, drugs are too large to pass through membranes by diffusion or transport. Endocytosis allows these drugs to be engulfed by the cell membrane, forming a vesicle that brings them into the cell. Example: insulin-loaded nanoparticles can be taken up by cells via receptor-mediated endocytosis. 5. Efflux transport. Efflux transport involves the active expulsion of drugs from cells, usually through transporters like P-glycoprotein, PGP. This mechanism can limit drug absorption and contribute to multi-drug resistance. Example: doxorubicin, an anti-cancer drug, is often expelled from cells by PGP, reducing its effectiveness. 6. Paracellular transport. Drugs can also cross membranes via paracellular transport, moving between cells rather than through them. This is typically seen in tissues with loose cell connections, such as in the small intestine. Example: magnesium ions are absorbed in the intestine through paracellular pathways. 7. Transcytosis. Transcytosis combines endocytosis and exocytosis, allowing drugs to be transported across cells and released on the other side. This mechanism is especially important for large molecules like proteins. Example: monoclonal antibodies are transported across endothelial cells via transcytosis, particularly across the blood-brain barrier. 8. Ion trapping. Ion trapping occurs when ionizable drugs are trapped on one side of a membrane due to pH differences across the membrane. A drug that crosses in its uncharged form can become ionized and trapped in a compartment where the pH keeps it charged. Example: aspirin experiences ion trapping in the stomach, where the acidic environment traps the drug in its ionized form, limiting its reabsorption. Conclusion: each of these mechanisms, passive diffusion, facilitated diffusion, active transport, endocytosis, efflux transport, paracellular transport, transcytosis, and ion trapping, plays a critical role in how drugs cross biological membranes. Understanding these pathways is essential for optimizing drug delivery and ensuring that medications reach their intended targets effectively.